Welcome to a series of conversations with people who help influence our world. Today we are talking to Dr. Subramanyam Vangala, who is CEO of Reagene Pharmaceutical and Biosciences Private Limited. Hello, sir. So, who is also a mentor for people who are in the Bangalore biotech ecosystem. Welcome to the show.
Thank you, Akash, for having me here.
So basically, what we want to understand is right now a lot of people are interested in clinical trials.
Right.
So, for a lay person, what is a clinical trial, and why do they have to be knowledgeable about it?
Yeah, a clinical trial is for drugs. Basically, what you're trying to do is treat a disease with a drug that has a positive effect. May not necessarily for cure, but at least subside the symptoms and makes you feel good. So this is what you need to know is that drugs also have a lot of toxic effects. So you need to find a balance between the safety and the efficacy of the drug to ensure the drug actually has benefit over risk ratio rather than risk over benefit ratio. So that's very important in a drug to be released into the market. To ensure that it is safe enough but works very well for the patient.
And going from there, I think that essentially is the process of drug discovery, right? But if you have to narrow it down and probably define it very carefully, how would you define it and how would you expound on it?
So there are two things, the drug discovery is a different ball game. It's a long process. I've seen drugs probably in my lifetime and never made it to the market, or some drugs made it to the market within 7 years, and some of them, and the average time is about 12 years for a drug to come out into the market, because there's a huge regulatory process that we need to be complying with to ensure that the patients are safe. That's the primary concern. Anything can be taken, but if it is not safe enough, you cannot put it on the market. That's the reason you have all these trials that you have. We also start with the preclinical trials, which is called phase 0, and then we go to, once phase 0 shows that animals are safe with very high doses, and then we actually take that data to predict what happens in the clinic. You go with the safest dose possible in the first phase 1 clinical trials. Phase 1 clinical trials are not supposed to be efficacy trials. They're only safety trials to make sure, given long enough, multiple doses, the drug is safe in the human before you actually do these trials are done in healthy subjects.
So this is toxicology test for healthy patients.
Healthy subjects. I would say I won't call them patients because they don't have any disease. But if it is safe enough in the healthy subjects for a period of time, whatever if the drug is efficacious over 2 weeks or 3 weeks, that period, if it is safe enough, then you immediately go into the launch into the phase 2 trials, which are basically efficacy trials with a limited number of population. You don't have too high population. Then if the drug has some positive effect in the small phase 2 clinical trials, you go into the phase 3 extended trials with a large number of populations. In some diseases, you don't even have 100 patients. But if you take heart disease and diabetes, etc., you have in millions of patients. So these clinical trials are done maybe in 4,000 to 5,000 patients. Just so the clinical trial design and the patient recruitment also depends on the number of patients available to recruit. So in some cases, recruitment itself takes 1 to 1.5 years. The trial may be only a 3 months trial, but recruitment itself can take 1 to 1.5 years. So these are the things that take a long time to be able to do entire drug discovery development program, all the way from drug discovery to the market, to convince the regulatory agencies that you have a very good drug that will treat particular disease, but at the same time it's safe enough to be given into the patients. So this is a long winding process and very costly, and 60% of the budgets go to the clinical trials. Before that, it's only about 30 to 40% you spend. But 90%.
What do you spend the 40% on?
Doing discovery chemistry, medicinal chemistry,
Basic
biology,
research,
pharmacology,
fundamental
to
research.
develop an animal model and see how the drug works. And then you also have to do preclinical safety trials in 2 species at least, in rats, maybe dogs and monkeys. So depending on those data only, you can nominate a compound to the clinical trial. It has to be safe enough at least in 2 species. So if I take rat and dog, and it is very toxic in dog but safe in rat, then I may not be able to take it to the clinical trial unless I show 2 species. So I also sometimes recruit monkey studies to be able to show that it is at least safe in rat and monkeys, not in dogs. Dog is not an ideal species for the human relevant species. So we actually, if the monkeys also trials are very good and safe, then we take it to the clinical trials. So this decision-making process is long and winding. Sometimes
How long are we talking here?
some drugs take 15 years to market.
15 years, what is the starting point of 15 years? So from the time you have the molecule concept?
Yeah, from the concept I'm talking about. So I have one target and say, hey, this is a very novel target. Nobody explored it before. And I want to take this, see if I have a drug that affects this target and make sure that that particular disease, maybe cancer or diabetes or whatever the disease endpoint you're looking at, and does it help in that disease. So the target is very essential in selection. And then what you do is you design drugs around the target. So the medicinal chemists come in and they actually design to see whether the chemistry is optimal to synthesize chemicals that will modify the target to have a positive effect. Once that is done, then you take into the animal models, and if the animal models show positive effect, then you go into the preclinical safety trials, animal trials. These are regulatory. FDA, ICH guidelines, EMA, they have several guidelines on how do you do these trials, and these are all protocol-driven studies, and make sure that you do them by the book.
By the book.
Yeah.
And these are essentially regulatory, civil regulatory agencies,
Right.
which kind of put
Right.
the guidelines.
Right. So if you think you have to modify these protocols and customize to your needs, you have to get pre-IND meeting with FDA to or regulatory agencies to make sure that if they agree, regulatory agencies agree. Oh, okay, he cannot use monkey or dog. He needs to want to use hamster or some guinea pig or something else. We need to get a regulatory blessing on that to make
Okay.
sure that before you do those animal trials as well. So these are the things that we need to be able to convince the regulatory agencies that we do by the book, because if regulatory agencies do require safe drug into the human. That's their primary objective.
Yeah, protect
Right.
consumers from the harmful effects if
Right.
they may.
So I'll give you one example here. You've seen the saga with hydroxychloroquine in the last
Correct.
10 months.
It's been in the news.
Yeah, it's been in the news for the COVID treatment. And the last 10 months, you've been seeing a lot of doctors and clinical trials are done. And everybody says that hydroxychloroquine is a good drug. It has some positive effects on the COVID disease. But FDA didn't agree to it. And FDA first gave emergency use authorization. After a month later, they revoked it. The reason they said was that because most of the people who are taking hydroxychloroquine also have comorbidities with other the disease. Those drugs may interfere with this drug and have severe side effects. The primary one is arrhythmias.
So hydroxychloroquine has comorbid effects with arrhythmia patients. Meaning
Not
like what?
arrhythmia patients. I mean, if I'm diabetic, if I'm taking metformin, just giving an example.
Metformin, yeah. Okay.
Taking metformin, and if metformin and hydroxychloroquine interact with each other, the hydroxychloroquine or even metformin go to higher level drug concentrations that may impact your heartbeat.
Okay. So these
are the reasons why they say that. Another reason I actually wrote a review article on this. that just published last 2 weeks ago in Pharma
Okay.
Bio, just showing that if you give a single dose of hydroxychloroquine, half-life is 22 days. I mean, it lasts forever. So if you want to reach a steady state by giving daily doses to hydroxychloroquine to humans, it will take 6 months to get a steady state concentrations. So, which means that it is accumulating in your body.
Okay.
And somebody else already suggested with the lupus patients is that you
Lupus
don't give
Pharma,
daily,
yeah.
you don't give daily doses. You give a dose once a week.
Okay.
That way you don't have this accumulation problem and you cover for the safety. And these are the things that doctors also should know to ensure that they shouldn't be giving the drug every day. They should be giving based on the pharmacokinetic parameters like half-life, volume of distribution, other things. So these are the things that determine how efficacious or how safe the drug is in the patients.
And you talked about interaction effects of drugs.
Right.
So what about hydroxychloroquine? Consumption of hydroxychloroquine without any interaction effects.
So hydroxychloroquine, in my article I also wrote that there are quite a few drugs interact with hydroxychloroquine. Many times hydroxychloroquine actually inhibits those drugs' metabolism or some other part, and their drug concentrations go up. And they are the ones who are causing. For example, azithromycin is there. Azithromycin is very well known a cardiotoxic drug. Initial trials with hydroxychloroquine were done with azithromycin.
Okay.
And this is where everybody said it's working very well. Azithromycin is just an antibiotic.
Okay.
It's not an antiviral drug. Hydroxychloroquine is also not an antiviral drug, but it seems to have immunomodulatory effects, whereby it may inhibit the cytokine storm. It's a passive effect, but it doesn't directly inhibit the virus or kill the virus, hydroxychloroquine. So both of them together have side effects that are really life-threatening. And this is where FDA came in and said, hey, we have all these clinical trials done. I think I've seen 24 or more clinical trials on the hydroxychloroquine itself. But the FDA final conclusion is that hydroxychloroquine is not safe enough to be given to the patients. Please do not use it.
Okay. That's why they, removed the emergency authorization
Right,
that
exactly,
they gave.
yeah.
So right now, are they? Is it part of the therapeutics life, cycle? Meaning, like, are they prescribing that for patients who have this COVID disease?
So for the COVID disease, I mean, there are lots of drugs out there. Our own Biocon, Torozumab is there, and many other cytokine inhibitors. These are all the drugs that these protein antibody drugs actually inhibit some of the cytokines that are elevated during the viral infection. Any viral infection has inflammation associated with it.
Inflammation.
So they stop the inflammation or reduce the inflammatory response to be able to, for the patient to be able to feel free. Because the inflammatory cytokines also have harmful side effects.
Okay.
And that actually is a double whammy for a virus because it's actually increasing the cytokine storm, which is supposed to kill the virus or stop the virus from getting infecting more and more. But these things come and kill your own cells, basically.
Okay.
Cytokines.
Okay.
So they don't discriminate between the virus and your normal cell. And that's when the inflammation actually becomes compounded effect and the patient goes into a much more serious condition.
So, and the thing that we have to understand, I think there was one term that we initially referred to but did not quite define for the layperson. There is usually a target for a drug, before you conceptualize something in the lab. There is a target. So, what do you mean by target for the layperson to understand?
In fact, traditional drug discovery back until 1980s and 1990s is called a phenotypic approach. Phenotypic approach means you know symptoms of the disease.
Correct.
And you give a drug and see if those symptoms disappear or subside. So basically that's how you call it phenotypic. But the problem with symptoms is that 10 diseases have the same symptoms. So you don't know which disease you're controlling because you may not be treating the right disease symptoms. So the problem is diagnosis is very important, right? So
Correct.
for diagnostics, there's some genetic defect or a protein defect or something else biological defect is there. Identifying that biological defect is called target identification.
Okay. And this is in the body of the patient.
Yeah, in a patient or
It's a population.
disease, yeah. And that target is the crucial one that you're targeting and say that, okay, inhibiting this target or activating this target, either way can subside the disease based on the biochemistry and molecular biology of the disease process. And what you're saying that, okay, I have an effect on this particular target on a outside, you take the target, put it in a test tube, and test your drugs and see how well they bind or inhibit or functional assays also in cells. And then you come to a level that my drug is actually having positive effect on this
On this
target. And then what you do is you want to design more drugs around it to make sure that you have a more positive effects than side effects because you're looking at the biochemical parameters and other modulators in a cell systems, unicellular systems where this target is overexpressed. And then you say that, okay, there are some biomarkers, we call them. If the target is modulated, it creates some biochemical effects around it. So those biochemical effects spill out some biomarkers, we call it, either a protein biomarker or a RNA marker or something. And you can use that biomarker to say that my disease is getting modulated. It may not be treated, but at least my drug is having an effect on this target. It gets more complicated when you go to the clinical trial. These are easy to do in cell systems, but in patients you cannot do so easily. So when you go to the patients, you don't even know where the drug is going in your body. So there are lots of innovative clinical trials, very expensive models are there that pharma companies are employing in the US right now to use the microdosing technology where it is the lowest safe dose possible that you administer to the patient and see there are some labels that you radioactive labels, specialized radioactive labels. You put it on the drug and see if the drug actually reaches the target by actually imaging analysis.
Okay, so the idea is you take the drug, you isolate the target, you check the effect of the isolated target afterwards through imaging.
Right. So what you do? What you do is you're actually imaging the patient.
You're making the patient.
Imaging the patient.
Oh, imaging the patient. Okay, got it.
Right.
Yeah.
You're.
I mean, how often? I mean, you take the drug today. You take the image the next day, or how often do you have to image?
Yeah, it depends on the label that you're putting on the drug. That labels, many of these labels that we use on these drugs are very, very short-lived.
Okay.
So you have to do it immediately or.
As soon as somebody takes the drug, you just put them in an
Right, and see where it is going in the body. That's it. Where it is going in the body. You want it to treat Alzheimer's disease, it has to go to the brain in specific locations. And image brain and see if that drug is there or not with the label through an imaging process.
We'll get to Alzheimer's if we can. So I think one of the questions that had written down was, aren't there enough drugs? Which seems like a tautology in COVID world because COVID is a nice scenario which shows the world that there aren't enough drugs. So that claim is sort of self-explanatory now these days.
Right.
What do you have to say on that?
Yeah, I mean, see, to go into the disease, apart from our regular diseases like diabetes, and we understood very well diabetes, maybe atherosclerosis, we understand very well. We have enough drugs around. Antibiotics, we know, we've got tons of them and everything. The problem comes to is that rare genetic diseases. There are more than 6,000 rare genetic diseases in humans.
Okay.
This is where the available drugs today are not sufficient to be able to treat all these variable 6,000 rare. For example, you take autism in the kids, right?
Okay.
I read about a few years ago that autism is not a single monogenic disorder. It's not a single gene disorder. There are 800 different mutations were detected in different organisms.
So there are 800 genes, they all interact and they all express or suppress themselves, and
There are mutations, point mutations.
okay.
800
Changes, yeah.
different mutations. The same gene could have 10 different mutations, or
Yeah.
10 isolated. You may have one mutation, I may have another mutation. Like that, there are 800 mutations were detected that predispose these kids to autism disease.
So what is the pathway for a challenge as
So
complicated as
this puts a lot of pressure on the research R&D programs. Because now you're not looking at a all or none effect, right? So what you're taking, you're not saying that all these 100 patients are the same anymore.
Yeah.
Each individual patient may, out of 800 mutations in 100,000 population of people, you have to divide and say, hey, 20 people may have this mutation and 20 other people. So you have to devise a drug for each 20 patients. You cannot have one drug to treat all these 10,000, 100,000.
So essentially personalize it as much as possible. And
absolutely.
The
that is where we are coming to. Yeah.
And the sample size becomes smaller and smaller because the recruitment of a very specified targeted group is hard because they're all spread out everywhere.
Right.
If they're spread out everywhere, the cost of recruiting is higher.
I'll give you one, just nothing to do with drugs. I'll give you one example. Cigarette smoke. We know it causes lung cancer.
Yes.
You know how many people actually get lung cancer? Less than 20% people who smoke cigarettes.
Okay, even amongst the people who smoke cigarettes?
Smokes, chain smokers, maybe 2 packets a day. I've known a lot of friends who've been doing that for the last 40 years. They don't have any symptoms of cancer or lung cancer. But the only 20% of people get cancer. What is the biology behind them getting that cancer, lung cancer, as opposed to those people who don't get cancer? This is the trickiest part because, out of those 20% people also, there are subgroups again. Some people get interstitial lung cancers, there's some mesotheliomas. So the cancers are also different in this field. So that makes it harder to nail it down to what is the biological effect of a drug or anything that you're trying to treat these cancers.
If we thought the thought of drug discovery was tough enough, looking at the COVID situation, the picture that you're painting with quite a broad brush seems to scare anybody who even thinks about this, because the R&D challenges given this scenario can be extremely difficult in terms of how you recruit patients, how you recruit people for clinical trial, how do you ensure that your recruitment processes are such that, obviously you randomize it enough,
right,
but you have to still look for people who would be conducive for the study.
Right.
And it becomes very hard given your scenario.
So there's a disease called a Pompe disease. It's a glycogen storage disorder disease. So
Okay.
kids who are born with that disease, most of them die by age 15. Some survive, but it's called adult Pompe disease. But anyway, the science from a lot of other companies in the US and everything, what they did was they cloned the gene for this enzyme that is deficient in these patients. And what they did was they expressed it in vitro in other cellular models, mammalian cell models, and synthesized this protein. And then they put it in the bottle. The patient can get injections.
Okay.
In India, I looked at how many patients are really there. So far, I think 20 to 25 patients. Now, that American drug, to come and give it, I think the cost is more than $100,000 per year. Here, I think so.
100,000 a year. That's
I think so, if I remember correctly. I got to go back and look at the numbers, but it's a very expensive drug. That families are putting their entire
life earnings.
livelihood onto treating their children.
Yeah.
Okay, so that expensive. In India, importing the drug and treating these patients it's impossible. And this is where the science has to discover more mechanisms on how to treat these diseases cheaper way rather than doing, and we are trying to do that. We've got some ideas on that and how we can develop a smaller molecule drug repurposing. There are drugs out there in the market, as I said, there may be 3,000 to 4,000 drugs are already on the market. One of them should be able to work for these glycogen storage disorders. We are looking into it. Hopefully we'll be successful coming out with a drug that may treat with a 10 rupees rather than 10 rupees a day. You give it a lifetime for a patient, that's nothing compared to $100,000 drug to treat a patient. So these are the things that to be innovated to be able to reach out to the patients that really need these drugs.
This will be a nice time for a plug for the company, right? So Reagene Pharmaceutical and Biosciences. So what does Reagene mean?
So there are different angles to it. In fact, we conceptualized this idea back in 2006.
Okay.
Myself and two other friends. The other friend who lives in Hyderabad, he was a former CSO, Dr. Uday Saxena of Reddy Labs, and he, myself, and another gentleman who's a serial entrepreneur. He just filed a company for IPO in the US, and we actually conceptualized this idea. We were actually roommates in Canada while doing PhD.
Okay.
Okay, so
all three
of us. We were all roommates in the same room, and there was another physics graduate student who was there, but he's not involved in biochemistry, life sciences research area. But now we come together after nearly 40 years, and we said, what can we do with our experiences put together?
Hence, the moral of the story is treat your roommates carefully because they will become your partners. So, and what does Reagene do?
So the conceptual, we three actually met in 2006 sometime in New Jersey at my home, in fact. And at that time we conceptualized an idea, just sitting there at the dinner table, and we conceptualized it. We had a couple of meetings, but we couldn't be worked out because we all have daytime jobs. Full-time daytime jobs, right?
At the time you were working with,
conceptualizing this idea.
and you were with J&J at the time?
I was at J&J at the time. Satish was an entrepreneur, and at the time he was working for a company called Nucleonics, his own company.
Okay.
And Uday was full-time with Reddy Labs, who was in Atlanta, Georgia, built their drug discovery programs in the US. So we all had our own daytime jobs. None of us were able to diverse our jobs, right?
Correct.
And that's pushed the timelines. And in 2016, I approached the Saxena again. Let's start it.
And the one-line idea at that point was?
The idea was to create reagents and based on the need. For example, in India we import a lot of reagents, biochemical reagents. Right, they're all expensive. If I have to buy something, it will take 3 weeks to come, and I have to pay customs duties and also other fish shipping charges and everything. In fact, in the US, I used to get the same reagent next day on my table when I ordered it.
Okay.
Here, I forget the idea by the time that reagent reaches to my.
So you forget the idea for which you ordered the reagent.
This is the problem. One of the things that we need to solve the problem in India. In Indian context. So we're building reagent database in India, make in India for India reagents. That's for the primary concept. Okay. Now but we are not looking at reagents as such biochemical reagents that are available in Sigma or anything else. We are into specialty reagents solutions company. So what we are trying to do is, right now, our company has evolved into that concept stage into 3D bioprinting tool reagents.
3D bioprinting tool reagents. Okay.
So they have different applications. One is for drug discovery, one is for toxicology, one is for patient trials. They have 3 different models. Our idea is to be able to develop these proprietary tools and patent them and license them out.
Okay.
Okay? If you want tomorrow that you want to use my tool, pay me and I'll give you the tool. And you can do your own research in your own lab with that tool. Whatever the way you want it, customized to your needs. Or if you want to outsource it to me and do it, I'm not big proponent of SC as a CRO, but I would like to see if there's any collaborative programs between us that we could actually benefit together.
So
Either a revenue sharing model or something else we could think about it.
So the end product is a scheme or a reagent?
So a tool. It's basically a tool that is we conceptualized it and brought it to that market. And that tool can be very useful for a pharmaceutical company. It can be very useful for a CRO company, another CRO company. It can be very useful for a toxicology company, industrial toxicology company. All of these can use this tool. Okay. And the tool may be 100% perfect, but we can fine tune it for your need.
So when you say a tool, you're talking about a software tool?
This is not software. We are also going that way. So what we want to do is actually, this is right now we're doing it in terms of collaborating with Tech Mahindra, and especially on the COVID program we're doing it.
I think you announced it a few months back.
Right.
Yeah.
So we are also looking at bringing the artificial intelligence tools.
Okay.
And combine with our 3D tools. As I said, 90% of the clinical trials fail in the humans.
90% of the clinical trials fail in
the humans. So as I said also that 60% of budgets are spent on the clinical trials. So if I fail in phase 3 trial, imagine I already spent close to $1.5 billion.
$1.5 billion with a B, meaning close to 10,000 crores.
Right. So you put them into the drainage. That's it. You got to the drain. If your phase 3 trial fails. So the question is this is the question we asked. How do you improve the success rates of the clinical trials? I said 90% fail. Can you bring them to 50% failures? Means 50% actually success rates.
And the way you think you can do that
is because animals are not capturing the human physiology. Okay. So animal models that are being done, these are regulatory mandated studies. Ours is not a regulatory tool yet. We plan to make it a regulatory tool one day, if possible. But it's a right time to do it because there are also a lot of animal ethics issues are also involved. Just killing the animals for no drug is coming out is also an ethical issue, basically.
Yeah, correct.
Right?
Yeah.
So, for that reason, if we can bring a tool and show to the regulatory agencies that our tool is better than animal tools that can capture. We're not saying to regulatory agencies that get rid of the animal trials. Maybe you can prioritize our trials first and only do the animal trials that are minimum required. But EPA, Environmental Protection Agency in the US, has already promised by 2035 they'll completely eliminate animal studies by using 3D bioprinting tools.
They have said that?
They've said that. They already gave a public
announcement. So animal testing is completely gone by 2035?
Exactly. They promised it. So this is kind of research that we need, but what is missing in this toolbox is the reagents that predict the human outcomes.
Okay.
What I mean by reagents is that if you take a liver, right, human liver, liver has 5 different cell types.
Yeah.
They all interact with each other in the liver.
Okay.
If I put one drug and put up one enzyme there, the cascade goes on, you don't know what's happening in the liver.
Okay.
Liver. That you cannot study with an animal liver.
Understood. You understood. So
how do we get it? So there are different ways of creating these organoids. People are doing it now. What we are doing is a 3D bioprinting. Organoids, spheroids, 3D bioprinting tools. What people are doing is they're combining these 5 different cell types and creating an organoid. Or they're differentiating stem cells to liver-like organs. Okay. So there are different ways of doing this, but everybody's idea is to be able to create human-like liver.
Human-like liver
that I can work in outside.
In the lab.
In the lab.
Yeah. And how many companies are there which are doing something on those lines?
In the US, there are lots of companies. I can't even count US and Europe. There's one company called Emulate that has made miles. Right now they're even working with FDA to actually make this model toxicology relevant model for human development. Because as I said, many drugs fail in the clinic due to safety issues. Primarily is a liver safety issue. The secondary is the kidney issue. Not kidney, I mean heart issue, and kidney issue third one. So they're primarily creating for that reason, can they reduce or minimize the liver toxicity failures by using this liver organ on a chip model, and they're collaborating with the FDA to be able to screen a lot of compounds. You know, you and me only can look at the literature on the PubMed and everything that we know what's published. A lot of companies don't publish their data outside. That data is actually with FDA.
Correct.
Right? So if you collaborate with FDA, FDA is going to give you the drugs that you don't even know that exist and died because of the hepatic failures.
Meaning like there are some private markets that, you know, these are private companies which are submitting to the FDA, so they are
right, exactly. But they don't many times give the reason why their drugs fail.
Yeah.
That information is only with the FDA available.
Okay. So collaborating with FDA is one way by which you can mitigate your prospective.
Yeah, because the FDA knows more than what we know outside in the literature.
Yeah, understood. So just to understand, take a step back and understand your personal journey. Like you went to Canada for your graduate school, right? So
right.
The motivation to go to Canada was obviously at the time. This is
1981. I left.
1981. So at that time, US was like a land of opportunity. Let's say North American continent was like a land of opportunities, and people who were pursuing science, specifically let's say biology or chemistry, were going there in droves. But you were part of that whole, let's say, group of people, or like you thought like this was an interesting thing for me personally?
Okay, I think I have to go back to 1979.
1979. Okay.
1979, I actually finished my MSc in biochemistry in Andhra University.
Okay.
I was clueless, I tell you, like anybody else at that time who graduated with an MSc degree, I was clueless.
Okay.
Don't know what next. But the only thing I know that I want to stay in biochemistry. That's the only thing I knew. Many of my seniors and others, they went into IPS, IAS, IFS, and all the civil services, and also bank probationary jobs and everything, because there were no jobs in biochemistry.
Back in 80s,
There were no jobs. There were absolutely no jobs at the time in India. You get degree, but don't know what to do with it. I came to IISc here in Bangalore for an interview. I was not selected.
Okay.
I mean, it's not me, but they have their own way of selecting people. And I was not worried about it. I went back and I went to IIT Delhi in the biochemical engineering department for a PhD interview. I'm not an engineering guy, I'm a biochemist. So they asked me some engineering questions I could not fare well, so I could not get there either.
Okay.
Then I saw an advertisement for a senior scientist job, some scientist job in CFTRMI, so I applied for it. My classmate got it. Now he just retired last year, but I didn't get it. So these are all the things that happened to me. I finally took UGC scholarship in Andhra University and stayed there. But my ambition was telling me that I need to go somewhere. So I was walking on the road, and my old friend, who was just leaving to Canada to do a PhD, but he's a statistician. He got his bachelor's degree in ISI Calcutta and MStat in Delhi. And he said, I got the Waterloo PhD program. So that prompted me to actually go and look for universities in Canada and the US in the library. I used to spend 4 to 5 hours in the library and getting their applications and applying in the US. I didn't have GRE at the time. Okay.
Was
so,
that a mandatory thing for applying to GRE?
For many universities, it was a mandatory thing.
Okay.
But again, some people were giving me admissions, but no scholarship, no nothing. If you have your own money, you can come over.
Okay.
But my father was a professor in the university. We were not I could not afford to go on my own. So I kept writing, right? And there was one university finally in Canada, Newfoundland, St. John's. Suddenly a professor writes to me, Peter J. O'Brien. I mean, that's why sometimes...
Which university is it? I didn't catch it.
Memorial University, St. John's, Newfoundland, Canada.
Okay.
That professor wrote to me saying that I saw your application, you want to do cancer research. My lab is doing cancer research. If you're interested, I'll submit your application to the Dean of Graduate Studies and get you $6,000 scholarship. On top of it, you can do teaching assistantships and I can also pay you some from my own research grants.
So you got a complete scholarship.
Scholarship about $10,000.
Okay.
My ticket to Canada. That was it. I never had to look back.
Correct. So when you land there, then you meet Uday Saxena, your co-founder.
No, not immediately. What happened was I was working already at Peter O'Brien's lab, and this was January 1981.
Okay.
And he comes there at May 1981.
Okay.
That's when he worked for a neighboring lab, Professor Mukherjee, who was the head of the department of biochemistry at the time. So that's how we became friends. Satish Chandran, as I already described to you, was working in the molecular biology department in School of Medicine. And
okay.
He also joined in May 1981. We became roommates.
Okay.
Long-term friends for nearly 40 years.
Yeah.
Okay.
So you finish your graduate school and then you go
So I got a postdoc offer in Toronto, University of Toronto.
Okay.
But I looked at it, even Canada doesn't have many jobs, to tell you the truth.
Okay.
I mean, this is reality. I mean,
Yeah,
you got to do the reality check. You got to.
What is the population of Canada in 1988?
Maybe at that time it was 22 million. I think it's about 38 million or something.
Now, yeah.
But there were no jobs. I mean, tell you the truth, I could become a postdoc for life there. And I've seen a lot of people just stayed as postdocs in Canada. And I said, there's not many industry there. Pharma industry was really non-existent there.
Yeah.
So I could live my life as a postdoc, and if I become lucky, I can become a professor there in the university. There are not that many universities, about 20 universities.
Yeah.
So luckily, one professor from Colorado who read my publications, a very nice person, he also called me up and said, hey, I read your publications, I have a postdoc position, I advertised in Science. There are 75 applications, but you didn't apply for it. I want you to come and join my lab.
So you did not apply. The professor called you and asked you to apply through this.
And he took me for an interview, and then right away he offered me a fellowship.
That was so nice of him.
Right.
Yeah.
But sometimes life is luck. I'm telling you, you have to be at the right place at the right time. And he gave me a 2-year postdoc position. Okay. I don't know what I'm going to do at the end of 2 years, right?
Yeah.
I went to a conference. I was presenting a poster at toxicology conference. There was this gentleman comes to me and says that, are you Manny? They called me Manny at the time. In Newfoundland, they used to call me Superman. That's another story that I will tell you later. But that guy, he's a full professor. He's not even 30 years old at Berkeley.
Okay.
Not even 30 years old. He's got 350 publications on his track record already at that age.
Okay.
And he approaches me and he says that I was ready to explain my poster to him, right? He was not interested in listening to my poster. And he started talking to me. So when is your postdoc going to finish with David Ross in Colorado? Maybe end of December. We're looking for recruiting people on a non-tenure track level right now. Like a research assistant professor, not funded by soft money, but in 3 to 4 years, if you can get a grant of your own, you can become a full-time assistant professor in Berkeley. But I want you to think about it. I'll recommend to the head of the School of Public Health, and we'll recruit you into the School of Public Health.
School of Public Health, okay.
University of California.
University of California, Berkeley.
Then I just went there and they sponsored for my green card and everything I got. I wrote 2 to 3 grants and none of them got funded, unfortunately. So the industry started knocking on my door.
And how long was it? How long was your tenure at Berkeley before you thought
I joined in 89 January in Berkeley. I left 92 September.
Okay. So in 3
Little
years.
close to 4 years, little
Okay.
close to, yeah. By end of the year it would have been 4 years, but yeah. But anyway, industry started knocking on my door.
What industry? I mean, obviously it's pharma, but which was the company?
Pharma, American Cyanamide, it doesn't exist anymore.
Okay.
You heard of this gentleman that discovered antibiotics, anti-cancer drugs, very popular Indian guy. I forget the name right now.
It's okay. We'll put it up anyway.
Right.
So you got but you were leaving Berkeley for a private job. That also means like you're giving up on your, let's say, academic
interest.
aspirations. Right?
It was a difficult decision to make. I was looking at benefits and risks of leaving the Berkeley job. I may never get a grant of my own if I stayed back in Berkeley.
That's also
right?
true. Yeah.
That's also, I need to weigh it out.
Yeah.
Whereas industry is offering twice as much salary,
Yeah.
and they're also giving a lot of stock options, bonuses. So I had to make a decision, and where do I want to be? Opportunity came to the door. If I leave it, I don't know when I'll get another opportunity to do it. So I took a quick decision in 1992 September. I moved to American Cyanamide as a senior scientist.
Okay.
And luck again, there was another gentleman who I was not reporting to, asked me to solve some clinical problems for a compound. It's in the clinical hold.
Okay.
So with my biochemistry, molecular biology knowledge, I solved the problem, and FDA withdrew the clinical hold, and they went back into the clinical program.
Okay.
So this gentleman, I met him in 1992 December, and I worked with him till 2007 sometime. Okay, it's almost 15 years I worked with him. He moved to Purdue Pharma. I moved with him as a group leader, and he moved as a global vice president to J&J. I moved him as a site director at J&J in New Jersey.
So you were his protege, so to speak.
Absolutely. I mean, I think I'm lucky to be associated with people who like to work with me, and I would like to work with them. It's a kind of chemistry we had together.
Exactly.
And which benefited me a lot and benefited them too. It's a mutual beneficial. We worked good together basically for 15 years, me and Dr. Alfred Tonelli.
So this entire tenure ensured that until 2006 is when you thought like you should start a company of your own. So what made you think that? Where the opportunities at company as big as Johnson & Johnson were not ripe for innovation inside the company?
Yeah, so Johnson & Johnson was also trying to move a lot of operations to China. Many companies were doing it at that time because China is cheaper, and also there's a lot of money going into China. So as anybody else, even J&J was thinking about it, and in fact, in front of me there were about 500 medicinal chemists and biologists were laid off on one day. And I realized that when J&J is not going to stay on the same course of drug discovery for a long time, they may move their operations elsewhere. So at that time I decided to move out, and I moved to Shire Human Genetic Therapies. But at that time I got calls to move into India to build infrastructures. One company picked me up with Cytochrome Life Sciences right now. It is called Advantium one time. They picked me up to build their infrastructure for biology, pharmacology, toxicology, biological, primarily DMPK. Build their infrastructures for it. So then I moved on, came to Advinus, and they already have the infrastructure to nurture the people and bring them to the speed of what's happening in the industry. So I actually managed the teams at Advinus also. So I built J&J's ground up DMPK Advantium steam here, ground up.
Yeah.
Brought them to a level that it's self-sustainable. But I have to move on in my life.
So these tenures were essentially training wheels for you to start your own company.
Absolutely. I got confidence at one level. And in fact, in 2012, I was actually thinking of starting something. My better half knows me more than anybody else. And she said, you don't have business acumen. Do you think you can do it?
Okay.
That's very important. You need somebody to tell you what's the weakness in you.
Correct.
Right? And
Did you
who
talk
can
to
better than your better half?
Yeah, exactly.
Right?
Did you talk to Uday at the time?
At that time, I didn't talk to Uday. Was still in Atlanta. Was working on second generation company with Dr. Reddy's. So he was still tied up with his own commitments. So in 2016, when I talked to him, when I came out of Advinus, he was ready. So that's how long it took. Because I didn't want to start on my own. Because you need to have partners who are also like-minded and who can tell you what you're doing, what you're doing right and what you're doing wrong. You need those kind of minds along with you. This is kind of a constructive criticism as well as appraisal.
Exactly.
You need to be aware of it. Don't take anything criticism personally, and make sure that there's something right in this, or I can take that advice and make sure that I don't make that mistake. So if somebody can analyze themselves, what is the self-awareness basically is. It's very important in life. And if you don't do the self-awareness and you think you know it all, you'll never succeed in life.
You can go off the course, and before you know, you're just about to drop from a cliff.
Right. Yeah.
And there's nobody to stop you. And worst of all situations, you're accelerating towards that end of the cliff.
Right.
So it might happen. So it's a good idea to always have partners in.
Who will give you the best advice that you can take, and that's how you have to live it. And in life, you take advice from people and see what is good for you and what is not good for you, and make informed decisions.
Were there any cultural shocks from, let's say, India to Canada, Canada to US, and let's say, vice versa?
Yeah, initially you'll have culture shocks, but I do believe the phrase that, some people say that, the saying that be a Roman when you're in Rome. So you have to adapt to new cultures. I think I've done a good job of adapting. Wherever I go, I adapt to the conditions. This is very interesting. In 2007, that Alfred Tonelli, who was my prodigy, I told him I'm planning to go back to India. There's this Indian guys who want to start a toxicology company, want to bet on me about 40 to 50 crores to start a company in India. What do you think about it? He gave his honest opinion to me. It's not that I don't want you to go back to India, but I'm asking you, how long have you lived in the US and Canada together? 28 years to date, I said to him. So what do you know about India? I said, I go once in 2 years for vacation for 1 month, but does it tell you how the business runs in India? So, you know what? Yeah. My honest opinion is that don't take this offer because you'll fail miserably.
Okay.
It was honest opinion. If you want to go still, take a job first, learn the know-hows in India.
Okay, don't bet on your own money.
Money right now. Right? I was not bidding on my own, but somebody else is bidding on me. But again, I think these are the things that you need to take it. You have to analyze it and say, yeah, there is a truth in this. They're not criticizing you for not becoming an entrepreneur, and you have to realize it and make sure. And that was a good idea he gave me because I came to India took a job inside Advantium. They built their infrastructures. I met a lot of people, and I understood India is different from when I left in 1980.
How is it different, and what are the key, let's say, pointers that someone who was in the same situation as you, who wants to, let's say, move back and either jump into entrepreneurship because he is a subject matter expert, or for that matter, he just wants to move back because it's a better cultural fit?
Uh,
What are the key things that you have to remember?
I don't know if it is advice or just my viewpoints are that, I mean, I would love Indians to come back to India. I mean, I came back, I made a decision, came back. I never regretted it. Okay. And the same thing is that you come here and keep complaining about India and this and that, you'll never be able to assimilate yourself. But on the other hand, if you actually adapt to the Indian culture and make use of it, turn it to your side. The adaptability is one of the key ingredients in your life's success. If you don't have the adaptability, you can never succeed. And so the adaptability is very important when you make a decision to move to another country or come back to your own country. That you cannot compare. You cannot compare India to US anytime. I've done that, and I don't do that anymore. But the thing is, it's very important. Sometimes you also have to voice your opinion to make sure that you give the right direction for the India also to grow to allow. Because I've seen best of the both worlds there and here. People are gems, no doubt about it. Indians are gems. There's no doubt about it, no question about it. And what we need is the education system needs to improve. Okay. I've seen in the US, I struggled for the first 1 year because I never had any practical bench experience.
Okay.
That gave me a hard time because once you go into a lab and try to do experiments, and you have to be thinker. You've got to be thinking of solving the problems, not creating problems. You've got to solve the problems. Solve the problems is a different ball game compared to creating problems. You can create hundreds of problems, but if you don't solve the problems, then the problems will be there forever. So what it gave me, the training gave me for 1 year, is that I need to be able to solve the problems, not to identify more and more problems.
Yeah.
And that kind of practical training is lacking yet in Indian education system. I mean, there are many reasons. There's nothing wrong about the Indian education system. I survived it. I'm glad I got my basic education in India. Right?
And you're glad because?
I mean, glad because actually gave me fundamental foundations of education, although not a practical applicability for it. I learned the practical applicability in other countries. So now I'm bringing that here to India to be able to teach the younger generations practical applicability of knowledge.
Okay.
Right?
Yep.
If you don't practically apply that knowledge, you can get nowhere. Yes, everybody has ideas. You have tons of, I'm a think tank. I can come up with 100 ideas. But the problem is I can't work on every idea that I have in my mind.
What is feasible, what is
You've
commercial?
got to focus.
Yeah.
I can walk and say to her, hey Akash, this is the idea, that is the idea. But so what? Right?
Exactly, yeah.
But so I need to say, okay, this is the idea I need to focus on. That is how Reagene came into existence. Because finally I thought there is an idea that I can actually capitalize on, make it on. So the ideas keep coming, let them come in, but see the merits and demerits of each idea you have, and see is it going to be successful or not. For that you need to do a lot of background research.
Yeah.
And if you don't do that, you'll waste your time. So you do your background first to make sure that this idea will definitely catch in the next 5 years to take it to the next level.
And you have to only do sure things, right? You have to be absolutely sure.
Right. So, focusing is very important. This is what director of American Cyanamide told me. Very nice guy, and he said, "Hey, Manny, you have tons of ideas, but you don't focus on anything." Right? That struck me. Oh, focus. You gotta focus. If you don't focus, that you're putting your hands in 100 different things, but nothing comes out of it.
I don't think any known person has pulled off building his own company or inside another company wherein he has his, let's say, time distorted by 10 things or even 5 things. So what Buffett says on this subject is if you have 100 ideas, forget the 98 of them, probably do 1 or 2.
Yeah.
And that's the way you can even get anywhere in those ideas. The 98, people do not have enough focus, enough time to do all those. So you can only do one.
And also money, you need
money. Absolutely.
I mean, where's the money coming to? I mean, I just read somebody posted our biotech group 2 days ago. The 10 top startup companies in India today. You look at all of them, majority of them, I won't say all of them, majority of them are actually doing genomic-based platforms for the personalized healthcare. That's where the money is. MedGenome raised $200 million in that. Farcast raised 70 to $80 million on that. So Anu Acharya raised, I don't know, but maybe $2-3 million. I don't know. I mean, so if you look at that, the wave today is a personalized cancer genome. You need IT industry and the scientific industry to actually come together and build these databases to treat the patients with a personalized care rather than one size fits all treatment. So, I mean, back in olden days, this drug is available. Okay, he's got cancer, give this drug. But 98% of the patients, the drug doesn't work.
So getting back to let's properly, precisely define these stages of clinical trial. So the first stage is?
Phase 0, we call it, the concept going into regulatory preclinical development. You select the 2 or 3 good molecules that you have, and they have very good safety profile, compared to the efficacy profile. So you have a better efficacy profile than the safety issues. So you take those molecules into preclinical toxicology in animal trials. These are regulatory mandated trials. You even dose animals at 2,000 milligram per kilogram doses, and I've seen that, and I don't know the rationale for it, but these are required mandated studies based on. But today we actually apply the bioanalytical tools, actually understand the concentration in the blood and the tissues, maybe the response to the efficacy as well as toxicity, and see if it is a narrow therapeutic margin or a wide therapeutic margin. So safety is here, efficacy is here. So you have a lot of therapeutic margin. Even if I give a little overdose, still it's safe.
It's
right?
safe.
So that's very essential in the preclinical toxicology, that you establish wide safety margin. Where the trick is that you have to establish the dose to go to the first in human clinical trials. The reason is because in animals we take mouse, rat, dog, and monkey. We do species related pharmacokinetics, we call it. And this pharmacokinetics will tell you how a mouse is different from monkey, or monkey is different from dog, or something like that. Because you are using 4 different animal species here.
Correct.
If there are differences in that, you'll know that there will be major differences in humans too. So the pharmacokinetics is very linear and doing very well, because mouse is 20 grams, rat is 200 grams, monkey is 3 kilograms, dog is 10 kilograms. So human is about 70 kilograms on the average. So you scale it up and say, hey, my allometric scaling data is suggesting this dose to go into the humans for an effective dose. But the effective dose, we don't know if it is safe or very toxic, we don't know.
Does it linearly translate in terms of weight?
There are lots of ways to do it: body surface area, body weight, brain weight. People do different kinds of ways, but typically standard procedure is body weight. But I've done it, I think it would be 30% times I'm correct, 70% time I'm wrong.
So even a subject matter expert will go wrong, and somebody who's planning the study has to take that into account when he is budgeting for it.
Yes. This is where the informed clinical trial design comes in. Your design of the clinical trials depends on this data. Correct. It's not just checking a box that I did a dog toxicology, monkey toxicology, and rat tox. But how do you use that to design a human-informed clinical trial?
To sort of customize and learn
from it, and say, hey, this is what I need to do. Go into the first in human safe dose, right? So, so that's the first thing we do. And the toxicology studies done. In the toxicology studies also you're doing pharmacokinetics, we call it toxicokinetics. You now know which dose is toxic.
So this is phase 1 basically.
Phase 0 in animals.
It's still in animals, okay.
Yeah.
Phase 0.
So you get the data saying that, okay, this is the toxic dose, this is the blood concentration I have. The effective concentration is here or here. Basically, that you calculate it. And if the effective dose and the toxic dose doesn't have different margin, we say this is not a good clinical candidate, we don't go anywhere. But, so having said that, this concentration response is very essential in designing a clinical trial. And on top of it, the other thing we do is that humans definitely pharmacogenetically different. When you're doing animal trials, rats are kept in a cage. They have the best AC room, germ-free room.
Okay.
Humans don't have that.
So you're not isolating the candidate or the subject?
In humans, we all eat different food every day. You know, you eat pizza one day, another day you eat
something
chaat
else.
masala. So animals are given the same food.
Okay, there's more uniformity in...
So all these variables affect in humans.
Okay.
So when you're trying to take, to extrapolate the animal safety data, efficacy data, you need to take these into account when going into the clinical trial. So human metabolism is different.
Okay.
Okay, quite different. We don't metabolize like rats or dogs or humans, monkeys, sorry, in humans.
Okay.
So there's an enzyme called Cytochrome P540. It metabolizes about 30% of the marketed drugs today we have in the market. 30%.
Okay.
So there's another enzyme called Cytochrome P453, A4 in the liver, that metabolizes about 50% of the marketed drugs today. So together these two account for 80% of the marketed drugs metabolized. So if I'm developing a new drug, if it is metabolized by these two enzymes in the humans, and if you're taking the other drugs are also metabolized by CYP3A4, you're prone to have drug-drug interaction, serious side effects. This need to be also tested in the preclinical level.
So you're saying if you're coming up with a new drug, you have to look at the broad array of drugs which are already in the market and how many drugs for
how
which...
they interact with the new drug that I'm developing.
Correct.
Okay, so that's very essential. Number two is that these are cytochrome P540 enzymes. There are other enzymes, the transporter, drug transporters. A drug needs to be transported. Wherever it goes in the body, there are transporters, uptake transporters and efflux transporters.
Okay.
They take it and deliver it to the other side. If these transporters are different from person to person, the drug doesn't work. Some work only in few people, it doesn't work in other people. That's what we call it pharmacogenetics.
Okay.
About a dozen of us from various pharma companies. In 2008, we published a pharma white paper on pharmacogenomics. I was one of the co-authors on it.
Okay.
To show that CYP2D6 enzyme is very crucial in understanding, because CYP2D6 is an enzyme which 30% of the patients are metabolizing. So we do CYP2D6 polymorphisms. There are 100 different mutations in human populations in different ethnic groups.
When you take that drug.
Yeah, so there are, if you take some Caucasians of European descent, CYP2D6 may be about 5 to 7 copies.
Okay.
So they metabolize the drug 100% in fraction of a second.
Okay.
So your drug never reaches the target, active site, to be able to be effective. It's not toxic, but it doesn't work in those patients who have 7 copies of that gene, CYP2D6 gene. On the other hand, there are some people who have, about 10% of Caucasians are deficient of the enzyme, because there's one single cytochrome P540 2D6 star 4 mutation, they call it.
Okay.
And that mutation is there. There's no cytochrome P540 enzyme. The enzyme is dead. There's nothing is there. So you don't metabolize the drug. So now your concentrations are way up. This guy doesn't have any concentrations. This guy has this much concentration.
Yeah.
It's very toxic for these people.
Okay. Okay.
So pharmacogenetics becomes a very useful tool when you go to the clinical trials. And this is where the industry is trying to understand the genetic differences among the humans. Forget about animals. You and me are different.
Yeah, absolutely.
There's no animal model to define you and define me.
Yeah.
Right? And this is where the problem comes in selecting an animal model to define. So this is where a personalized medicine, precision medicine, is the talk of today.
Okay.
So we are, as Reagene Biosciences, building these personalized medicine precision tools to be able to work for you and work for me. So what I can do is go to the doctors and say, hey, this person has some cancer or some other disease. You want to treat him, but you don't know which drug you want to give him, because you're giving one drug, it doesn't work. You're giving second drug, it doesn't work. You give third drug, it doesn't work. So you don't even know which drug works for this patient.
Correct.
Right?
Yeah.
And you delay another 6 months like this, giving 10 different drugs, the patient is going to die anyway. So for us, if you give us a biopsy or even blood sample of this patient, we can understand what is the difference between in this particular patient. We can tell you which drugs to give the patient by using our in vitro human 3D bioprinted tool.
Okay.
So we'll put his patient biopsy cells into our tool and screen the patient. What is he deficient, or what is he having high levels? So we can actually do the genotyping, but also see which drugs actually work for him, and we can give a combination or a single drug, whatever that need. And doctor can now precisely give that medicine to make it work for that particular patient.